Well, thank you very much indeed for that build-up. I hope we can live up to it. And thank you all for coming. So in these inaugural lectures, we're asked to speak about top-class science, but we're also asked to include in the narrative our own experiences and as it were, build in the story of our own careers into the science narrative. And that's what I'll try to do. And my title here is deliberately ambiguous. Okay, so there's two kinds of inner space that we're going to be talking about, if you like. So one is the inner space inside our cells, and I want to talk about machinery, the walking machinery, which actually moves around inside the space inside our cells. And of course, the other inner space is the inner space of our own experience, and that's exactly also the thing that I'm going to try to incorporate. Interweave into the narrative that I'm going to give you. So let's get started. So how do we do this? So what I want to say essentially is that our bodies are composed of machinery, and the machinery that moves things around inside our bodies is of special interest. This is what I'm interested in. And it's what my colleagues are interested in, and it's essentially been the theme of my research career so far, the machinery that moves things around inside our bodies. And the reason that this machinery exists is because the problem of organising a human body, or indeed the problem of organising a single cell, is very considerable indeed. Okay, so here's a single cell. This is a skin cell. Now this cell is about 0.1 millimeters across. Okay, so this is a very large cell, and it's large because it's spread itself over the surface that it's attached to. But in this typical hypothetical human being here, we have this many cells, about 10 to the 13 cells. What this human being has to achieve, if he's going to continue to function correctly, is to get all of these cells in the right place. And to get all of the contents of all of these cells in the right place at the right time in order to sustain function. So we're going to be talking about machinery that achieves this inside the cell. So here's our single cell. What you can see is that the space inside the cell, that's one too many clicks, there we go. The space inside this cell is filled with a system of tracks. These are called cytoskeletal filaments, and they constitute a kind of railway system inside the cell. So the cell needs to deliver packets of molecules, molecular ingredients for the machinery that it wants to build, and it has to deliver those accurately and at the right speed and to the right destination at the right moment. And this machinery that moves along these tracks in order to deliver these packets of ingredients or components is absolutely fundamental to the life of the cell, and equally it's an extremely useful thing to target with therapy, with medical therapy. So let's just take a journey now in our imagination into this region that I'm going to call inner space. Imagine that you could, in your mind's eye, sort of plummet into an individual cell. This is roughly what you would see. If you're going in through the membrane of the cell, this is what you would see. So these are individual filaments of a substance called actin. This is actually also found in your muscles, and I'll be talking a bit more about muscles later on. These filaments assemble themselves, and this is an important property of all of this whole system. They're actually well capable of putting themselves together without external direction. So this is this web of actin filaments which just sits directly underneath the cell membrane. The cell has a whole battery of tools and other little machines for manipulating the arrangements of these filaments. This is one that severs them. It also has a lot of material for cross-linking those filaments and that kind of stuff. It also has a system of a different style of tracks, and these are called microtubules. And these microtubules also assemble themselves, just using the information that's embedded in their individual component molecules. This is not something that's directed from the outside. This is something that's a property of the proteins that make up these tracks. And as you can see from this cartoon, the tracks come and go cheerfully the whole time. And they're tracks in the sense, as I said, of railway tracks. So along these tracks, you have trafficking. these little machines, this one's called kinesin, which walk like a tiny human being along this system of tracks. And they pull, like Atlas, enormous great packets of molecules contained within these membranous sacs. These are called transport vesicles. And in fact, these same molecules move much larger cargoes like the nucleus, lysosomes, ribosomes, all those kinds of things that you will know about if you've studied cell biology. So that's the message. We have inner space inside the cell. It's very large compared to a molecule. And in order to get things to move around correctly at the right time, to the right place, at the right velocity, the cell uses this army of transport machines that step along these trackways. And that's what I've been interested in, essentially, for my whole career. So that was all a cartoon, in case you didn't realise. That was a fantasy. This, on the other hand, is the real thing. So this is microscope data from my lab. This is what you see if you take cells from a living creature. And this is actually this is cells from a rat liver. You take those cells and you break them open. You actually break the membranes and you allow the contents of the cell, which is called the cytoplasm, to come out and to spread around onto a glass microscope cover slip. And look at that with appropriate optics. This is what you see. These individual strands here are the individual fibres of this track system which exists inside the cell. These are the microtubules. And you can see that along these microtubules you have a great deal of trafficking of these transport vesicles. The thing to appreciate about this is that we haven't done anything special. This is just the material that gets exuded from the cell, slightly purified by what's called density gradient centrifugation, but not otherwise interfered with. So these things are moving under their own power along these tracks, just using the fuel which surrounds them ordinarily when they're in the cell. And they're moving at quite high velocity. So this distance here is perhaps about 50 micrometers, or about 1/20th of a millimeter. And you can see that these vesicles are moving at a large fraction of that every second. And at the speeds that these motors move, these walking machines move, it would only take you a matter of about a minute or so to walk across an entire cell. These are fast, and this is room temperature. And these data that I've been showing you, this is real time. It's not been fooled around with in any way. So the question is, how do you get a handle on the problem of how this works? This is machinery in many ways in the ordinary sense. It would be as if you were looking at a car engine or a railway engine or something, and you say, well, I just want to understand how this works. So what do? How do you actually go about figuring out how it works? And of course the problem is it's very small. I mean, it's extraordinarily small. These tracks that I've been telling you about, the microtubules, are about 25 nanometers across. That's 25 thousandths of a millimeter across. And the individual steps that these molecules take as they move along the tracks are even smaller. They're only about 8 nanometers. So it's an extraordinarily difficult problem. You cannot see. This is about what the light microscope can do without very inventive kind of technical innovation. How do you actually get down to it? And the substance of what we do, which we could call biophysical cell biology, or as we recently renamed it, mechanochemical cell biology. The substance of what we do is really it's really involved in getting down to that question of how it all works. So this is a rough diagram of what is understood so far. So this is about 25 nanometers in diameter across here. These individual building blocks are about 8 nanometers long, and they plug together into these strands that we call protofilaments, which form up into a microtubule inside the cell, as I said earlier, under their own steam, as it were. Then this is a kinesin molecule, which has these two identical little heads, as we call them, which engage with the footsteps, the kind of footfalls, which are presented to the motor on the microtubule here. So these take little walking steps of 8 nanometers each, hand over hand along the microtubule. Then this is attached via a tether, and then at this end there's a cargo selector those are very variable because there's very large teams of these things inside the cell which carry different cargoes around, and the cell clearly needs to program which things move in which direction at which time, and that's the job of the cargo selector at the other end there. Now, so that's the problem. We need to be able to purify these proteins. We need techniques to work out exactly how they work as machines, despite the fact that they're only 4 nanometers across here, the actual business end. Extraordinarily small for any normal kind of mechanical problem. We need techniques to look at that. And we also, of course, need funding. And then the question is, well, why would you actually want, here we are, Warwick Medical School has just hired a large team of people who are interested in these problems. Why do we want to look at these problems in a medical school? This is clearly a sort of interesting problem. It's interesting in the sense that it's relevant to the whole problem of how life works. It's relevant to the whole problem of how human bodies set themselves out, but it's equally relevant to the problem of how a plant sets itself out, or something, or a nematode or something. Everything in nature, which has a nucleus, uses these techniques to organize the contents of its cells. So the questions that we're asking are very widely relevant, but specifically, why are they relevant to medicine? So if you just sort of box that off for a moment, that's the problem. Now, why are we interested? Well, we're interested because when you look in fine detail at how this microtubule is plugged together with these little subunits that I was telling you about, these are heterodimers of tubulin. And when you look in fine detail at the molecular structure of this walking device here, then the opportunity arises to start to design things that are actually going to fit into this structure and interfere with its function. And that's the reason that this is medically interesting, because if we can make suitable small molecules, drugs, which can recognize sites within these proteins, bind into them, and actually modify their function, then you've got a very, very powerful tool in your hands. And for medicine, it's a powerful tool. It's equally powerful for something like crop protection. There are fungicides which are based on specifically binding to particular kinds of microtubules, hitting the fungal microtubules, but not the plant microtubules, that kind of thing. In medicine, we have things like this. This is Taxol. This is a famously useful in cancer therapy, and increasingly in other conditions as well. What this does is it fits into the molecular structure of these subunits that make up the microtubule, and it locks it and prevents it from changing shape. And as a result, it prevents microtubules from reorganizing themselves in the way that I showed you in that animation at the beginning. So Taxol stops microtubules from reorganizing, and thereby stops cells from dividing, because cells need to reorganize microtubules in order to get into a position where they can divide, in order to set up the apparatus for moving the contents of the cells apart in cell division. Cells need to take down their microtubule network and rebuild it into a new configuration. And this drug, Taxol, blocks that and thereby blocks cell division. And it's extremely useful, therefore, in cancer therapy and in other situations where you've got too much cell division. And equally, on the side of the motor, we now have new families of drugs which are specific to particular classes of these walking machines. And you can add these to cells, and it shuts down the walking action of the motor, prevents this transport from happening along the microtubules, and thereby blocks, for example, cell division, but also other types of transport within the cell. So this is not a theoretical interest. These are actual useful therapeutics that already exist, and we know that this is a helpful way to go in terms of developing new therapies. So that's the medical connection. That's the reason that we want to look at these things, because we know that they're great things to treat with drugs. And just to emphasize that, here's a little animation. This is a microtubule that you're sort of zooming along here. These little wriggly things here are intended to look like tau protein. Tau protein is a microtubule binding protein, which is a real problem in Alzheimer's. So in Alzheimer's brains, there are very, very large amounts of tau, and exactly what relation to the original disease this overproduction of tau has is still very much a debate. But what's not at issue is that when you've got very large amounts of this tau protein around in the brain, it assembles itself into great lumps of tangles and sort of clots, and forms roadblocks which prevent this walking action of the kinesin motor along the microtubules. So the fundamentals of your brain require that there is continuous traffic of bags of neurotransmitter chemicals along the neuronal axons to the nerve termini. That's going on the whole time in your body right now. And it's going on in your brain to resupply the chemical transmitters which allow you to signal between the neurons in your brain and allow you to think. And in Alzheimer's brains, this transport of neurotransmitters, as they're called, is interfered with, and that interferes with the person's ability to think and remember. So we're very interested in the possibility also to use chemical agents to improve this transport along these microtubules. And just to generalise the point, there is, this is called discovery science, not basic science these days, but it's the same point. The idea is by discovering more about how this machinery works, we get to be able to design and develop better drugs. And then those better drugs, oddly enough, also allow us better to investigate the machinery that we're interested in. So there's a virtuous circle. The more discovery science we do, the better the armory of chemical agents which can be used therapeutically, and the better the discovery science gets round and round in that virtuous circle. Right, so the last thing to tell you about in this introductory section is what are we trying to do now in this new Centre for Mechanochemical Cell Biology, which Peter can now say.
Without his dentures.
I have to say the Vice-Chancellor cannot yet.
No, what can I say?
So the centre is not the building, the centre is the people. This is a very important point. And these people are the leaders of this new centre. So 4 of these people came from the old Marie Curie Centre, as Peter pointed out. So we have a history of working on this machinery for the benefit of cancer research, funded by the Marie Curie Foundation, Marie Curie Cancer Care. And the 5th member, Masanori Mishima, who's just joined us. I'm sorry, this is Andrew McKench, Anna Strauber, Nick Carter, this is me, and this is Masanori Mishima, who's just joined us. He's moved from the Gurdon Institute in Cambridge, and he's CR UK funded, Cancer Research UK funded. So we're very heavily funded by cancer charities at this point, and we're aiming to diversify. What the centre is trying to do is trying to understand these machineries which move things around inside cells, what we call motorised self-organisation in cells. That's the mission, and the connection to medicine I've already made. So that's what we're about. Now, that's the end of sort of the pitch, if you like, for the kind of science that we're interested in, and the questions that I'm interested in, and the questions which drive that science. What I now have license to do, because this is an inaugural lecture, is tell you my story. So now we're going to flip slightly to the other kind of inner space, the internal experience of a developing scientist, as it were. So I'm going to sort of take you through some of my influences, and I had to scratch my head about this because I don't often think about these things, but I'm going to now explain some of these influences, and we'll see how this goes. So here's one more. Everyone of a certain age, right? So this, I was 4.5. This is black and white television, ladies and gentlemen. We were fortunate in my house to have a black and white television because my father, who's here today, was a gadget hound enthusiast and still is, and he built one. So televisions were not widely available when I was 4.5 years old, but we had one, and this was profoundly influential on me. You can imagine just how exciting it is to see space travel at this age. And this is Steve's Zodiac. Here making the schoolboy error of trying to parachute in deep space. And of course, not very soon afterwards, but not very long afterwards either, came real space exploration. And this is an extremely influential experience in my life, and I suspect also in other people's lives, other scientists of my age. This is the real, this is the real thing. So we can't do sound, I'm afraid, today, but you're going to have to. Maybe I can do this a little bit.
Contact light. Okay, engine stop. We copy you down, Eagle. Houston, Tranquility Base here. The Eagle has landed.
So just an extraordinary thing, and tremendously exciting for I was 11 at that time, and I remember staying up all night watching this thing on TV. And it was the spirit of the age, and I think it's tremendously surprising to anybody who's around at that time that this exploration of space seems to have sort of died a death. I think it's a tragedy. And it's just slightly encouraging that the Americans are now, along with the Russians, because no individual country can afford it any longer, they're now actually trying to do the Mars challenge, and that I think that's entirely what the human race should be doing, rather than war. Should be exploring space, and that's extremely influential. So the other extremely influential thing on me was further gadgetry. So again, those of you who are as old as me will remember this. This is what a calculator looked like when I was 15 years old. And we learned, I thought this was pretty amazing. Just the fact that this worked seemed to be pretty amazing. But then into my 15-year-old life came an extraordinary thing. My father, who, as I told you, was a frighteningly kind of enthusiastic gadget hound, brought this home. I never did find out exactly how he came to be in possession of it. I mean, it cost a month's salary in 1973. But this was like an alien artifact, and it's just absolutely thrilling. This was straight out of what later became Silicon Valley. This was the world's first handheld computer, I think it's fair to say. And it was a business calculator, and my father was a management consultant. But there was a matching scientific calculator, and this thing worked in reverse Polish logic. It was a handheld computer that you could program. It's absolutely staggeringly exciting at that point, and my enthusiasm for gadgets hasn't looked back, really. So that's the end of that little personal part. Last, this is slightly more general, and again, people remember this. I think this is again when I was 15 years old. This is a BBC series, which I think is still on Parallel, but still available. You can get this on YouTube, or you can buy the DVDs or whatever. This is Jakob Bronowski, who was a member of a generation of physicists he was involved in the Manhattan Project, and like the other people who are involved, got horrified by the consequences of that. And he was commissioned, he did a lot of popular science programming on the BBC in the late 60s, including a radio kind of panel that you could phone into and ask questions of. He was commissioned by the BBC to make a program about the cultural history of science. And this is it. It's called The Ascent of Man. I just recommend anybody who's interested in the history of science and the cultural influence of science over the years to make the effort to go and find this, because it was extraordinarily influential on me at that time. You may have gathered I was a real television hound. What Bronowski contributed, essentially, to my development was just this sense that science is the most interesting thing which is going on in the world at the moment, and I haven't shifted from that view either. That might be seen to be quite a hard view, but that's nonetheless my view. So anyway, following all these influences, where I finished up was this, which is not very far from here. I scraped into university with the help of my bewildered parents, and I'm extremely grateful for them for sort of wangling me in here. I got in under clearing, and probably I shouldn't be telling you this because my son's sitting in the audience. And I just had the most amazing time there. I just had a lovely... My mother went there as a student as well, so there was a family history. And Sutton Bonington, it turns out, has developed enormously since then. This is the view on Google Maps, and it's twice as big as it was when I was there. This is the lab that we worked in. This is the hostel that I was in charge of. At one point, I was a hostel warden. Had a tremendous time and fell under the influence... Oh, this is me hurrying to a lecture. Only babies wore trousers in those days. I fell under the influence of this gentleman, who's also in the audience. Ron Bardsley was obviously made of different stuff to most of the people that one encountered in agricultural college. He was extremely interested in enzyme mechanism. He quoted at me all kinds of tremendously inspiring views. He said that enzymes were the cathedrals of the 20th century, as it then was, and I think that's probably right. So he's extremely enthusiastic, and he transmitted that enthusiasm to me and interested me in muscle. So Ron's interest in muscle, I think, is continuing. He's retired now, but he's still working, as I learned this morning, as indeed are most of my retired friends are still in the lab. Muscle is an enormous agglomeration or assembly of exactly the same sorts of machines that I've just been telling you about. So in order to move traffic around in a cell, you have to pick up a piece of cargo, a piece of molecular cargo, and step it along these tracks. In order to move your arm, you essentially use an extremely highly parallel array of similar machinery. So in order to pick up a kilogram, you've got about 10 to the 12, about a million molecules of this same type helping you do that. So then they're all working in sync, not in exact sync, but they're all working at the same time. So this is actually a piece of dead cow which has forgotten that it's supposed to be dead, and you can see it's contracting. And this is an individual muscle cell, and when the fuel is added, the ATP fuel that powers all these kinds of devices is added, this cell contracts, and you can also see that it's striped, and the stripes get closer together as it contracts. This is a single muscle cell. So that's what Ron interested me in. He's particularly interested in the molecules that were responsible for this contraction, and this is a cartoon illustrating them. This is the polymer of these little walking devices. This is myosin, as it's called. These are myofilaments, meaning they come from muscle. And these are the actin filaments from muscle as well. So what happens is this little motor hops its way along the actin filaments, and everybody hops exerts force in the same direction. And that causes the entire arrangement slowly to contract, or rapidly to contract if you're in a fast muscle. And the question that Ron interested me in, the question that he was interested in at that time, was if you look carefully now in detail at the surface of this polymer of myosin motors, and you think about the detail of exactly what's going on when this force is exerted on the actin filament. The question he was working on was whether this part of the molecule, which is called S2, actually desorbed from the surface of the thick filament, so that the head part here lifted away, as it is doing in this cartoon, lifted away from the filament, from the myosin filament surface, and then made this part of the molecule more exposed to the outside world. And specifically, the thing I got interested in was when this thing desorbs in this fashion, is it possible that it actually melts? So this structure, we haven't drawn this structure correctly, but just work with me here. So the idea would be that if you have a protein structure which is just marginally unstable, it's stable but only just. And then if you lift it away from the stabilizing environment here of the main part of the myosin filament, this would melt. And as a result, it was shortened. So that would go from that to that. And that shortening might be the origin of the sliding force in muscle. So that was the thing that we're interested in. I wanted to address that, and in fact managed to do so. We, partly in Nottingham and partly on a Royal Society funded trip to Salzburg, purified a lot of these, some of these proteins, not just from striated muscle, like the muscle you find in your arms and legs, but also from smooth muscle. like the muscle that you'd find in your bladder or in the iris of your eye, or that kind of muscle. That's very unusual sort of muscle without the stripes. And we purified molecules from both of these sources, and we asked about the melting temperature. So this was a biophysical question, and it was a particularly apposite question to be asking in Sutton Bonington because they were meat scientists. So it was possible to use the same apparatus that was used essentially to cook meat and decide whether it would go well in pet food and that kind of thing. It was possible to use the same apparatus to do this molecular study of exactly how muscle worked. We were able to show that, in fact, there isn't any melting, that in smooth muscle, this region of the myosin molecule was extremely stable, in fact. So that wouldn't do as a general concept for how muscle might work. So this really, I'm very proud of this work still, and it establishes quite clearly that muscle cannot be producing force from that region, or muscles in general cannot be producing force from that region. And that was enough so the product of my PhD work was enough to make it credible to apply for an EMBO fellowship to go back to Salzburg. And this is me in 1984, living down in Y in Kent, close to Y College, with my then girlfriend, Kim, who became my wife, and writing up a PhD on the dole, and waiting and hoping that this EMBO fellowship would actually come through. And so what happened was I eventually got a little job. I got a job in what was then a government agency called ADAS, the Agricultural Development and Advisory Service, and I was analysing the butter mountain for a living. And I got a call one day, and a voice said, "This is Hugh Huxley," and I thought, and I'm Cleopatra, but actually it really was Hugh Huxley. And so he'd been deputised to interview me for this EMBO fellowship. And this is Hugh giving a YouTube talk about how he discovered this the molecular foundations of this stripy arrangement in striated muscle. Hugh's still going strong. I talked to him last week in San Diego. Like most of my people, like most of my friends and acquaintances in science, he's not given up after retirement. He actually moved and took over an American university after he was retired from the MRC. So Hugh interviewed me for this fellowship, and I was absolutely terrified going to the MRC Laboratory of Molecular Biology and meeting Hugh Huxley at that time as the director and one of the most important scientists in the world, and interviewing little me. I was terrified, and Jake Kendrick-Jones got deputised to sort of calm me down by Hugh, and that also proved to be the foundations of a very rich ongoing relationship. I haven't forgotten that either. So anyway, we succeeded by some miracle, and I was translated to Salzburg in Austria. This is a picture of the Bischofsschloss, which is the huge castle of the Catholic bishop who used to govern Salzburg, and this is a view of the centre of the old city. This is the Spengler Bank, where I used to have to go on my bicycle to pick up my EMBO fellowship, amongst all kinds of black Mercedes driving, sort of ex-Nazi gloating kind of types who kept on turning up there as well. And I had a fantastic time in Salzburg as well. My mentors there were Vic Small, who's an amazing microscopist. He's also retired. He's also now running a lab in Vienna. Apollinaris Birschek, who's an extraordinary protein biochemist. And at that time, these two were best buddies, and I had the very privileged training from two of the best people in the business during this 2-year EMBO fellowship in Salzburg. Unfortunately, later on, they had the mother of all bust-ups that eventually shut the institute and caused a national scandal, but we'll gloss over that. The thing we were interested in was these single molecules of myosin. So myosin, remember, is the component molecular motor of these thick filaments in muscle. It's the thing that produces the force that allows you to do work with your body. We're interested in this molecule, and we're specifically interested in an unusual phenomenon which had just been picked up by another friend of mine, Hiro Inishi, in Japan. Inishi published a paper in 1982 in which he described something or other very mysterious happening with smooth muscle myosin, that under certain conditions it appeared to just close up on itself and jackknife and become much smaller. Now we know, these days, we now know considerably more about this, and what I'm showing you here is a paper from last year from Stan Burgess and Peter Knight, where they actually showed in molecular detail that this thing not only exists, but you can describe exactly which residues in the protein are responsible for stabilizing this folded up state. But in those days, we didn't know anything like that amount of detail, and the challenge was to try and figure out what was going on here. So, obviously I can't talk in detail about what we did for those 2 years, or indeed about the most of my career. But what I'm going to try and do is in each step we're going to just pick out a key result that I'll try and just run you through. So the key result, if you like, from this 2 years in Salzburg was discovering that when myosin folded up like that, it trapped radioactive nucleotides. So the fuel molecule that drives stepping got trapped in the active site of the molecule. And that's what this peak of radioactivity is here, that's coincident with this protein peak in this gel filtration experiment. And then when the molecule unfolded from that jackknifed state, then the nucleotide was no longer trapped. So there was a big clue here. It looked very much as though this folded up state was a kind of an off state of the molecule, and that there might be, and this was immediate apparent even right back at the beginning there might be the possibility that the myosin was moving between this folded up, turned off state and the assembled state where it's kind of built into the filament in the muscle and actually generating force. That has turned out to be correct, and I'll tell you a bit more about that in a moment. I did do a few more things in Austria. Learned to ski, did a lot more running up mountains mostly, and this is me on the top of the Großglockner, which is the highest mountain in Austria, on one occasion, very early in the morning. And just tremendous memories of all that, and I have a lot of friends in Austria still who we see. Anyway, this period in Austria gave rise, thankfully, to a number of papers, and this was enough to get me noticed, and was able to apply towards the end of that period in Austria for further fellowship, especially based on this trapping result that I just told you about. And was able to move, after a little flurry of uncertainty, to Cambridge to join J. Kendrick James, who's, as well as along with Ron Bardsley is in the audience, also J. Kendrick James is in the audience. And this is a picture of us running a meeting towards the end of my time at the MRC LNB. This, the problem, again, that we principally focused on during my time at the LNB was still this assembly problem, just trying to understand exactly what was going on there. You can see that this is the folded up version of the molecule, this is the straight version, and then this straight version can in principle assemble into the filament and disassemble again in this direction and so on. So I thought at the time that we did a pretty good job in various ways, not just me but several other people in Jake's lab, including Sandra Chiti. Pretty good job at ultimately sort of understanding how this is actually regulated and all the rest of it. But you can see that there's room to sort of revisit most of these things, endlessly. And this is a paper from last year. This is a diagram from last year, nearly 30 years after these original experiments. Still very actively investigated, but the story is basically the same. That in smooth muscle, this equilibrium is a reality, so that the contractile apparatus in smooth muscle is actually continuously redesigning and rebuilding itself. In these muscles, which are much more like non-muscle cells in many ways, the arrangements for generating force are much more fluid and adjustable than they are in a striated muscle. This is the Muscular Dystrophy Association that funded a fellowship for me when I was at the LNB. as did the Muscular Dystrophy Group in Britain. This is some of the things that we produced at that time. And I have to say that this experience of being at the LMB was just tremendous, because it was like changing gear in a very substantial way. I mean, the Salzburg Institute had been delightful, but it was relatively relaxed compared to the LMB, and the opportunity to sort of breathe the same air as genuinely great men and women was really something for me. So this is me with Aaron Klug, who's the director of the LMB at that time. Max Perutz was still there. He was the founder of the LMB. He was still there. Cesar Milstein, who discovered monoclonal antibodies. It was just an astonishing environment to experience, and a real privilege to have the chance to learn at the feet of these people. And other people too. So this is my mentor and collaborator, Clive Bagshaw, who, unique amongst my friends, has retired and appears to mean it. But he now lives in California. Clive is a very accomplished kineticist. He wrote the book essentially on myosin kinetics at quite an early stage in collaboration with David Trentham in Bristol, and that's also the origins of Ron Bardsley scientifically. Clive was distinguished for two things, really. He was absolutely excruciating coffee, which I suffered for 2.5 years, commuting backwards and forwards from Cambridge to Leicester to do these experiments. And also his equipment. So, as I'll show you, he had one of these, which was a really special thing. This is an Apple computer, ladies and gentlemen. But then, and we were able to make these measurements of trapping of nucleotide in the myosin active site and show that this was not something that just happened in this special smooth muscle case that I'd looked at in Austria, but also more generally. In fact, in striated muscle, in skeletal muscle, it also happens. So there's also this reaction occurs in skeletal muscle, and it's very likely, I think, that's important in muscle development. So the myosin needs a way of turning itself off in order that the apparatus can assemble without starting to generate force and mess everything up when it's assembling. So anyway, Clive had one of these, but Jake, here's Jake, who's also here today, setting an example of attitude, I think, which is really, really influential in many ways. Jake had one of these, right? So this I fell in love with totally. So this is one of the first, not this one, but obviously this model. It's one of the first sort of really friendly, usable desktop computers, and this appeared, and again, it just had a profound influence on me. These cost $6,500 in 1991. They were black and white, and they had a VGA screen. And I bought one out of my own money, which astonishingly my then wife tolerated. Gadget enthusiasm and gadgets driving science, I think, has been a strong theme in my science then and now. This was towards the end of the period in Cambridge. This is an EMBO workshop that Jake and I organised. You can see the dates of this. It was the 8th to the 13th of September 1990. And on the 17th of September 1990, other major things happened. So this guy is a lot bigger now. He's right here. And my life also moved on in big ways. So on the same, and in the next few months, did a job hunt and got several offers, but the one that we settled on was in the Marie Curie Research Institute, which was being run by Graham Curry down in Surrey. And unfortunately, Graham can't be with us. He's ill at the moment. But he recruited not only me, but also Carlos Cabrera from the LMB, and he was building up a really significant team in basic science in the Marie Curie Institute. This is Marie Curie. She's usually shown in later life when she's very much the worse for aware from the amount of radioactivity that she was exposed to during her scientific life. But she was beautiful to begin with, and she had a very beautiful mind as well. When she didn't say much, but when she did say things, they were along these lines. And I think the Marie Curie Foundation is well named after this woman, I have to say. So this is my first crew. I was able to move there with my baby son and wife, and we recruited these people. This is Isabel Cravel, Andrew Lockhart, Danny McKillop, and this is Maria, who's here today and is now my wife, and we'll get to that. Here's an early shot of the slightly later team. This is Nick Carter, who's already with me. This is Doug Drummond. This is Kurt Anderson. This is Maria and Isabel. The team gradually grew. This is us on the Seven Sisters on a retreat, and eventually you can't fit in the phone box any longer. This is the Molecular Motors group from, I'm not sure, that's probably about 8-9 years ago now. This is thinking about TB. And that's the final iteration. And most of these people actually moved to Warwick. So what was memorable from the Marie Curie experience? Well, so I'm just going to pick out two things from science from the Marie Curie Foundation, from the Marie Curie Institute. Keeping in mind that I was there for a very, very long time, as were my colleagues, for example, Jacob Dalgard, who moved with us from the Marie Curie. We had a substantial part of our lives with this. So this was one highlight. So this is a grant that we won in collaboration with several other people, including Joe Howard, who sort of piloted the whole thing. This is the Human Frontier Science Program, and this is an extraordinary, wonderful scheme which just gives sensible amounts of money to great science and doesn't ask for anything other than great science. And we were lucky that we got ranked first out of 256 applications for this. That was a really tremendous accolade, and the people on that grant are all still my friends. Brad and Linda are here today. Sharon Endow is at Duke University in North Carolina, and she's continued to work largely with Linda and Keiko on structural aspects of how kinesin micromotors interact with microtubules. Brad here has recently made FRS. He invented the confocal microscope, and he's now on our scientific board for the CMCB. This is Joe Howard, who's now director of Max Planck in Germany, and we collaborate with him still and pick up students from him still, and I saw him last week in San Diego as well. And is that everybody? That's everybody. So this is just one moment from the great thing about the Human Frontiers money is that you can use it for anything you like. So it's perfectly legitimate. Legitimate to travel to Japan, have a little meeting, and just discuss science and figure out what experiments you're going to do next with this tremendous international team who just all share ideas and inspirations and stuff. And you can see some people get into the swing of it and others just don't. So scientifically, aside from that experience of getting a grant, what did we actually sort of discover, which is interesting? And the thing I'm going to pick out is Nick Carter's work on single molecule mechanics of kinesin. So this is a diagram of the experiment that Nick set up. This is a single microtubule attached to a glass surface and just locked down very hard so it's not going to move. And this is in a light microscope, this arrangement. And then this is a single kinesin molecule attached to a plastic bead. And the reason we attach it to a plastic bead is because we want to have a handle with which we can manipulate this single kinesin molecule. The bead is transparent, and this is meant to be the beam, the focused beam of a continuous wave infrared laser. What that does is it gets focused through this spherical lens, effectively, of this little bead. And as it does so, the rays slightly change trajectory, and that imparts a tiny little bump of force to the bead. And when the bead's in the middle of all that, it gets held in the centre because those forces are symmetrical on either side. And you can use this arrangement to actually have a tug of war with this little kinesin molecule. So what it's going to do is the kinesin molecule is going to land on the track here, and then it steps 1, 2, 3, 4 away from the centre of the track, and it fights with this light beam, effectively. You have this working like a tractor beam, tending to hold the bead in the centre, and then the kinesin molecule tending to try to pull it away from the centre. And using this type of arrangement, Nick and I were able to figure out really very high spatiotemporal resolution the stepping action of a single motor molecule. This is the microscope that Nick built. This has now been translated here, it's now installed here, and we're working hard on the next iteration of this right now. This is us at the time. This is in 2005. This paper was published in Nature. It's been very highly cited. It's the first demonstration that kinesin, for example, can walk backwards under load, and it was the first time that anybody had really looked in fine detail at individual steps. And we were able to show that those steps occur on the same time scale for forwards and backwards steps, and that the half time is in the order of about 25 microseconds for that stepping action. So that was the Marie Curie. Now we're in the CMCB, and I want to just emphasise that the theme of the research that I've been telling you about will carry over very strongly to what we're trying to do here. We will work both on. We are working, because we've been here 2 years, and we are working on both the biophysics of the machinery inside the cells, which is delivering cargo to where it needs to be, which is manipulating the chromosomes at cell division, and doing countless other jobs of moving cargoes around inside the cells. We'll be working on the biophysics, and we'll be working on that as an engineering problem. We'll also be working on live cells. And there's a great deal of what we do is live cell imaging, to figure out whether what we discover in those in vitro experiments, in those experiments on the isolated apparatus, just in a rather unnatural kind of situation, an unnatural experiment, go back to the natural situation inside the cells and work out whether the same sorts of mechanisms apply inside the cells. And this is just a kind of montage of some of the things which we've done over the last couple of years to sort of give you a flavour of the sort of visual spectacle that's liable to come now out of this new facility that we just set up, which I've just been to a party this morning to kind of celebrate the finishing of the building. So it remains for me to thank various people for. Thank you, obviously, to Warwick Medical School for the opportunity to inaugurate myself, which is, thank you for recruiting us, because a very wise move, very good taste. Thank you to these people for raising me right, and they're here today as well. Thank you to this extraordinary woman who I have the privilege to spend my life with, who's also here today, and even if she's losing it a bit now. And thank you to my kids for not being too much of a pain. And thank you to the CMCB for following and challenging and criticising and just being generally an excellent group of people. Thank you for what they're doing. And finally, I'm going to leave this chap to have the last word, because I think that's the spirit of what we're trying to do, and that's the spirit we would love to transmit to other people here at Warwick. This is a privilege to be able to work on these problems. It's difficult, it's challenging, it taxes all the qualities of humanity. It taxes your patience, it taxes your physical dexterity, It taxes your ability to think in a creative way and in an analytical way. It taxes your self-discipline. But it's an extremely rewarding way to spend your time. And certainly recommend it to anybody who's got their stomach for it. So thank you very much indeed.